Hello everyone, I'm Dr. Carlos Rogério Figueiredo, an adjunct professor at the Faculty of Medicine in the University of Turku in Finland. Today I'm really excited to start our new series of lectures on pharmacology. The title of the lecture today is Drug Invention and the Pharmaceutical Industry. But bear in mind that this is just the part one of this lecture. Our goal today is to explore the fascinating world of drug invention and the pharmaceutical industry, which combines chemistry, biology, and even medicine. So, in today's lecture, we will take a journey from the ancient times when people discovered the power of plants and all the way to modern chemistry that we know today and how we find new drugs. So, let's dive in. This is an exciting topic. And I will start talking about the early experiences in human history with plants until the path that took us to the modern chemistry that we know today. So, humans have always been fascinated with finding ways to change how our body works, right? And often by using drugs in a good sense. So, this fascination started a long time ago when people, for example, realized that certain plants contain chemicals that can have effects on our bodies. Taking, for example, the story about an Arabian monk who discovered that the energizing effects of caffeine by simply watching goats play after eating coffee berries. There are also other types of plants, for example, Belladonna, which contains substances that can make us more beautiful and attractive, or can even be used as dangerous poisons. So even today, we still rely on plants for some medicines. For thousands of years, actually, the Chinese herb Mahuang, which contains like a substance called ephedrine, has been used to improve blood circulation in our body. So other examples, for example, in South America, the indigenous tribes have used arrow poison like curare for anesthesia, and we still use them in medicine today. Morphine, a pain-relieving drug that comes from the sap of poppy plants, and even though it can be addictive, it can be used in medical practice. So, as we entered the last 150 years, scientists, they start using synthetic chemicals instead of relying solely on the natural compound obtained from purification strategies coming from the plants themselves. So they discovered ways to create drugs in the laboratories. And this actually began in the dye industry, where some pioneers realized that certain chemicals, they could interact with our bodies and potentially be used as medicines. So this led to the development of drugs like arsphenamine or prontozil, which were the first treatments for infections caused by, for example, microorganisms. Since then, the collaboration between pharmacology, chemistry, and medicine has been crucial to develop these drugs and treating new diseases. And what are the source of drugs? So in the past, most of the drugs, they were small molecules made out of organic compounds, right? They could be a combination of different atoms of carbons with other molecules and atoms, and they would compose these organic compounds. But after the 1980s, we start a new era. It's a milestone in drug development that we start using now recombinant DNA to produce drugs that are based on proteins. So instead of relying on chance discoveries from plant extracts, for example, we now use high-throughput screening methods. And these screenings, they involve testing millions of compounds quickly using automated systems and robots even. So the goal is to find potential drugs that interact with specific proteins, the targets, or to produce a desired biological effect, for example. So even if that means to produce a new protein in a cell. So if the cells need to synthesize and produce or to have a product that has a biological function, you can use a drug to trigger this process. The problem is that the initial hits or the fragments of drugs that we are discovering to have these biological effects in the beginning, they might sometimes have side effects. So you need to counterbalance the effectivity and the side effects. So medicinal chemists, they create modified versions of these hits considering several factors like affinity, activity, absorption. It's very important to consider the absorption because you want this drug to be absorbed by the cells or even the tissues. So you need also to consider the molecular structure of a cell, which is pretty much composed in the surface by a double layer of phospholipids. So it's very hydrophobic, and the drug needs to penetrate that. So it needs to have some sort of affinity with a hydrophobic environment. So all these things need to be taken into account. And afterwards, the drug needs to travel through the body. So the distribution of this drug across different types of tissues and so forth. Afterwards, how this drug is transformed, because it will go to the liver and it will suffer transformation. So it's broken like in a different compound. So the metabolism of this drug needs also to be considered if you want this drug to be active after going through the liver, which is the organ that pretty much transforms everything. All the drugs are transformed in the liver. Most of them, they're taken endogenously. And then afterwards, consider the benefits. If you think that this drug is targeted the proposed protein to have its biological function, what are the side effects? Is this drug also able to induce a side effect or to interact with a normal or another target that you are not interested in? Eventually it can interact because they have a molecular structure that can have affinity with other proteins as well. So all these things are put in a scale in order to develop a new drug that will one day be in the market. So in recent decades, protein-based drugs, they're the most challenging ones to develop, by the way, because they are more complex molecules than the organic chemistry. So the protein-based drugs, they have become more important because they're more specific as well. It's difficult for these components or these structures and molecules to have off-site interaction with other targets, but it's also difficult to produce them. So we use a technology called recombinant DNA technology that we can produce large amounts of this protein in an easier way by inserting what we call a cloned gene with information to develop this particular protein. So if you look into the molecular biology and cell biology lectures and books as well, you will understand that one gene or one DNA, the DNA is composed of several recipes, that is recipes to form proteins. It has the information in its DNA code. So the genes, they are different recipes to create an RNA, which is the intermediate translation information to create a protein. So the protein is a different molecular structure compared with the DNA and the RNA. But the protein will start folding and creating these engines that will have functions. Most of the proteins that have an engine-like function, they are called enzymes. So these proteins, they can also be used as drugs. For example, hormones. Commercial hormones that can be obtained nowadays in the pharma and the pharmacy, they are protein-based drugs. Growth factors that can be used as well to induce the proliferation of cells and stimulating an immune response in a laboratory scale. Or even other substances, they have similar functions as well. So these are protein-based drugs. And how to optimize the chemical structure of a new drug. And remember, this is just an introduction lecture, okay? We're delving into the different strategies to create a drug and also considering the disease that we will approach, and we will bring a special focus here in cancer research. But in general aspects, how can you optimize the chemical structure of a new drug that you're considering development? So to understand this, we need to start thinking of a lock, right? Think of a lock and the key that you use to unlock this lock. So the lock represents a particular protein. And in our body, we have several different billions of proteins, right? So we have a lock that represents a protein, and the key represents the drug. The drug can be an organic molecule, that's a more simple molecule, or it could be also a protein-based drug. So the interaction between the drug, which is the key, and the lock, which is the protein in your body, what we call target, this interaction is crucial because it's not only about binding the key with the lock, it's also having affinity and function, right? It's not easy to develop a key that will open that particular door. You need to understand the structure of the key. You need to understand the structure of the lock and how they can have it needs to be a unique affinity. So then this machinery will work. So medicinal chemists, like locksmiths as well, they modify drugs to improve their fit with their lock, their key fitting with the lock. And obviously the interaction with this lock, which is pretty much the protein that we are talking about. So they use several new advanced techniques that are available nowadays. For example, the X-ray crystallography or even the nuclear magnetic resonance spectroscopy to provide detailed information about the drug, the structure of the drug, and also the target, the protein that they want to target, guiding these chemists in making adjustments to better fit the drug to that particular protein. So this is possible to do nowadays. And in the future, especially with the AI emerging now with new computational features and methods, they might play an even more important role in this process to make more stable and precise medicinal molecules with their even like improved functions in terms of absorption, distribution, and metabolism, elimination, and to minimize side effects. So we are living nowadays very exciting times to predict and design new drugs using the existing tools that are available. So with these tools, we can perform, for example, computation simulations. I can show to you here in a schematic, for example, some of these platforms, they are even online. And if you understand a little bit of organic chemistry and also the structure of different types of structures of a protein, we can already even see this like in practice. We could predict interactions using these models here. We can predict different structures of proteins and molecules that could interact with this particular pocket or points of this protein or target to identify promising molecules and compounds, and also to avoid unwanted interactions as well. This is very important. If you want to have a specific type of interaction in that molecule, or if you want to compete with another interaction you don't want to have, you can design a specific molecule that will bind in the middle of this cliff, for example, that will compete or prevent another molecule to bind there. This is how you develop new inhibitors or drugs that have an inhibitory function as well. So although this is still like, it's very like fascinating, right? But it sounds a little bit ambitious. We do have several advancements in computational biology and chemistry nowadays to make this more feasible. However, the recent developments that we have today, they shift towards the development of more advanced and complex structures and molecules to advance the therapeutic stages. So through genetic engineering, for example, we can produce human, or what we say humanized proteins, humanized because they are initially developed, for example, in mice, and then you preserve most of the similarities or commonalities between mice and humans, and you adjust the other differences as well. So you are humanizing a new protein, and you can develop these more complex molecules in large quantities as well. So this, I would say, custom proteins, they offer a certain advantage, like you can refine the design of these proteins, these custom proteins, by reducing the potential interactions that they would have eventually with our immune system. You must bear with me that we are talking about developing drugs and new proteins. These things are exogenous. They are not developed in ourselves. So we have a natural proclivity to react against exogenous stuff, right? We have reaction to pollen. Even like in the springtime, when we start like breathing outside, we start sneezing and develop all these immunological reactions. So pollen is just an organic substance, very complex, by the way. But we have a natural proclivity to develop allergy to things that are not proper from us. So these drugs, they need to be well refined. And it's a great advantage today with the existing tools that we can actually humanize proteins so they can be tolerable. So we can tolerate them. For example, the hormones that are developed in the labs, they are developed so and refined to not elicit immune responses against these hormones. We don't want to have allergy against hormones in a therapeutic setting, like growth factors, granulocyte, colony stimulating factor. We need those things to stimulate an immune response. Monoclonal antibodies. They're so used today, extremely effective for treating different diseases. It's like in the top therapeutic, standard therapeutic approach for treating cancer nowadays. Monoclonal antibodies, very specific to their targets. And other sorts of treatments as well to prevent autoimmune disease. So it's very important to note that these large molecules, they typically target proteins outside of cells. And they are usually given through injections, so they are dropping the blood. So it's highly likely, if they are not well designed, they can elicit a systemic immune reaction that could be lethal. So we don't want that, and that's why they need to go through a very refined process of humanization. And let's finally discuss today, it's just an introduction about targets of drugs, their actions, and the challenges that are existing today to understand the mechanism of action of this drug. So finding the right target is crucial for a drug. You can have multiple targets to have a final end. If you want to treat cancer, imagine the multitude of targets that are available that you would want to target, targets that we don't even know yet. So we're constantly discovering the targets, right, to achieve a final outcome, which is treating a particular disease in this context. So finding the right target is crucial. It's like finding a needle actually in a haystack. And it's important to confirm that the needle can indeed affect the disease. It cannot just be a noise in the middle of this mess. So questions start arising like during this process. For example, can the target be influenced by the drug itself? Has the target been validated as well? Is the target just a hypothesis, or you have a functional validation? No, this is really the target that I want to investigate. If I artificially neutralize this target somehow, or like if you block the expression of this target, that function is abolished. Or if you stimulate this target, you get even more of that particular function. So you need to study the target first. There's an entire period of science devoted to understand the target. And then you need to know if the development of the particular drug that will treat this target or target it, it's economically feasible, right? So the target is very important. It can be extremely challenging to develop something that would block or stimulate this target with the existing technology that we have today. Also, the drugability of the target depends on existing or having a binding site that can actually interact with the drug effectively. And specifically as well, it's not only you defining the target, okay, this is something extremely important, I need to neutralize this, or I need to stimulate this somehow. But then with the existing tools that we have today, or a plethora of different molecules and compounds that you use to develop a new drug, you find out in the end that nothing can bind this. Nothing can have affinity because the structure of this target is so closed, or it doesn't have like binding pockets. So the target is important, but with the existing technology and tools that we have today, it's not possible actually to have binding in this particular target. So you need to consider those things when developing a new drug. And even when we find a way actually to affect that particular protein or target, our body might eventually develop a mechanism of compensation to adapt to these new circumstances, make it even more challenging to validate that target with the new drug. So to overcome these challenges, we use what we call molecular biology techniques, such as gene manipulation and even animal models that we can use to study human mutations. But it's not just about the science behind of this. So drug development is also very expensive, which influences also the direction of this research. So all these points that I'm raising today, and I will continue raising the next part of this lecture, because this is just part one, they will create the solid grounds for this course. I will use these terms and all these challenges and processes in the drug development so you understand how to treat different types of cancer, for example. We will bring special focus here in cancer. I will also mention other diseases as well, but cancer is the main focus of our channel. And I want you to understand, in the end, I promise to you that you have a vast understanding of different types of drugs that are used today according with the needs of the patients, and also limitations in terms of availability and costs to treat these different types of cancers or diseases in general. Take into consideration of all these challenges involved in the drug development process. I hope you find this very exciting, because for me it's a very exciting topic in sciences, because it's our ability to treat the diseases, to manipulate our bodies so that we achieve better well-being and survival in face of the challenges of all diseases. So I hope that you enjoyed the first part of this lecture, and don't be shy. Share this with your friends, invite people to watch and join the channel as well. This is a fascinating topic that will unleash your mind to the understandings of medical sciences, and especially for drug development and how to treat diseases. I'll see you in the next video. Bye bye.